Hello, this is Dr. James Camp from Lee College in Baytown, Texas, and this is blood, the fluid tissue. Today we're going to talk about the general composition of blood, the composition of blood plasma, the functions of the formed elements, including the 5 types of leukocytes. We're going to talk briefly about hemoglobin, and we're going to talk about hematocrit. Okay, so what is in the blood? The blood is our body's main fluid tissue. It's not the only fluid tissue. There's also a fluid tissue called lymph that we'll talk about more in a future lecture. Um, but like any tissue, um, like any connective tissue, certainly, blood has an aspect of cellularity. Okay, it's about half formed elements, about 45% really. Um, and it's about half extracellular matrix. The extracellular matrix in this case is a liquid, which is what makes blood a unique connective tissue. And that liquid is called plasma. Uh, Blood is about 5 liters in the average person. The average woman has about 4 to 5 liters. The average human female, the average human male has about 5 to 6 liters. I'm not sure if that's entirely just because females, human females tend to have somewhat smaller body mass than human males, or if there's something about males that actually causes us to have more blood per body mass. Um, the blood has a very tightly uh, monitored composition. The temperature is about 38 degrees C or 100.4 degrees Fahrenheit. That may surprise you if you know that human body temperature, core body temperature, is about 37 degrees C or 98.6 degrees Fahrenheit. Uh, Blood temperature is actually slightly higher than body temperature, and this is used to help regulate our body temperature by circulating the blood to certain areas or away from certain areas. Uh, pH of blood is 7.4 plus or minus 0.05, so very narrow pH range. And that is a fact that we'll keep coming back to in a couple of chapters where we talk about the pH regulation of blood. That uh, on a scale from 0 to 14, this 0.05 tolerance is a pretty small number, and there are a number of mechanisms in the body that maintain blood pH within that. Okay, so another way of looking at what's in blood would be to take our blood, would be to take, say, about 10 cc's of whole blood and put them in a centrifuge for about 10 minutes. What you would come out with is a tube that had separated into a hard pellet at the bottom with a fluffy white coat known as the Buffy coat. Here I thought a Buffy coat was what you wore to slay vampires, but apparently it's part of your blood. Um, and then the liquid on the top of this is what we call plasma. Okay, the red pellet at the bottom, the deep red pellet at the bottom, is the erythrocytes. We'll talk about those in a minute. The Buffy coat is made up of mostly platelets by number, mostly leukocytes by volume. Okay, leukos is Greek for white, so these are your white blood cells, or WBCs in the typical clinical abbreviation. Erythros is Greek for red, so these are your red blood cells, or RBCs in the typical clinical abbreviation. Want to briefly talk about what's in plasma. Water is only about 92% of plasma by weight, which makes it a very thick solution. The the sterile saline that they will give you as an infusion at an ER or a doctor's office, or anytime that they're trying to do a intravenous, an IV infusion, is 99% water and 1% saline. Plasma, by contrast, is 92% water, 7% protein. Huge concentration of proteins in there. A lot of those are albumins, which help to maintain the osmotic balance of blood, but also serve often as carrier proteins that transport things that aren't very water soluble around the blood. There are globulins, including the famous immunoglobulins, also better known perhaps as antibodies. There is fibrinogen. Okay, fibrinogen is something that gives rise to fibrin, which is what blood clots are made of. Fibrinogen by itself will not form clots, and so it's safe to have 4% of it circulating around in your blood. But as soon as the clotting cascade that we'll talk about in a future lecture gets activated, fibrinogen becomes fibrin and forms a clot. And then there are regulatory proteins. What's a regulatory protein? Well, most commonly it's a protein hormone, a signal protein that's circulating in your blood for the purpose of regulating your body's response to something. Then the other 1% of your blood is other solutes. Okay. Now, other solutes, large quantity of this is electrolytes, sodium, potassium, chlorine, calcium, magnesium, phosphorus. So those are all electrolytes. Also HCO3 minus bicarbonate. This plays an important role in buffering the pH of your blood, keeping it within that very narrow limit that we talked about. Nutrients, so sugars, mostly glucose, amino acids, even ketones. You've all heard about ketones from the popular ketone or keto diet. Ketones are created in low carbohydrate conditions to give your cells that depend on carbohydrates usually something else to feed on. Feeding your body entirely on ketones is a sign of starvation, of carbohydrate starvation, and is not actually good for you for any significant period of time. We'll talk about that more in the nutrition section of this class. Respiratory gases. So there's a small amount of CO2 and a small amount of oxygen that actually dissolve in your blood. Most carbon dioxide is carried as carbonic acid. Most oxygen is carried on hemoglobin. And then there are waste products that are circulated in your blood until they can get to your liver or your kidneys. Most are removed by your kidneys. Okay. I want you to keep this snapshot of what's in plasma kind of in the back of your mind, because as we talk about plasma filtering through tissues to become lymph in a future lecture, as we talk about plasma filtering in the kidneys to become urine eventually, we need to have in mind what's originally in the plasma. What is it that we're filtering to get these other fluids? Now, I haven't zoomed in on these percentage numbers here, and you may have noticed that I skipped those. These percentages are representative numbers for a sort of typical human, but they're not always exactly those numbers. This 44% red blood cells that's indicated on this chart here is actually a critical measurement of the health of your blood, known as hematocrit. So adult males will have 42 to 56% packed red blood cells after centrifuging your blood. Adult females will have 38 to 46% packed red blood cells. Now, this is a difference between males and females. This isn't just that males have more blood. This is percentage. This is a measurement of just a 10 cc sample of blood. So hematocrit is the thing that we use to determine whether a patient is anemic or not. So there's actually a different threshold for anemia if you're male than if you're female. So if a male patient walked into the clinic with a hematocrit of 40, we'd say, well, that male is anemic. If a female of the same, general characteristics walked into our clinic and had a hematocrit of 40, we'd say, oh, she's fine. So the female has to have a lower hematocrit before we call her anemic.
What's a hematocrit?
So keep in mind that these numbers also have upper limits, and these upper limits should not be ignored either, because you can get a situation where there are too many red blood cells in your blood, and they start actually kind of clogging up your arterioles and capillaries. People who have tried blood doping, giving themselves hormones that artificially elevate their red blood cell count, which is a common practice among people who are training to be athletes, apparently. People who have tried this have sometimes even died from having an excessively high hematocrit. Okay, so let's take a look then specifically at these formed elements. Now, first off, why do we call them formed elements and not just cells, blood cells? I have two theories about that, hypotheses, whatever you want to call them. Two ideas. One is that we started studying blood before we started studying cells properly, and so there wasn't a nomenclature for this at the time, and formed elements just kind of stuck. The other reason that's more current is that the most common cells in blood, erythrocytes, red blood cells, are not actually cells. You can see that these erythrocytes look like little kind of pink jelly donuts in a way, and that is because they are missing something that most cells have. They're missing a nucleus. They're missing any other organelles at all, period. Okay? So if you imagined a normal human cell and you took all the organelles out of it, you might imagine it collapsing down to look like a red blood cell here. It's basically a sack full of the protein hemoglobin, which carries oxygen and a small amount of CO2 around the body. But before we get to hemoglobin, let's talk briefly about the shape of red blood cells here for a second. So these are called biconcave discs. Okay, concave means caved in. Okay, so a biconcave disc is one that is caved in on both sides. Why would this be an appropriate shape for a cell whose main job is transferring oxygen into itself in the lungs and then out of itself in tissues? Well, this gives more surface area for transfer per amount of stuffing, if you will, inside the cell. So if you picture a arbitrary oxygen molecule, the distance it has to travel to get outside the cell and then travel to some tissue that is around the cell is much smaller this way than if we had a big round cell and the oxygen molecule stuck here has to travel all the way to the edge of the cell. This also makes these the red blood cells, the erythrocytes, very flexible, so that as they're passing through small spaces known as capillaries, they can bend and flex and make it through those small spaces without destroying the cells. Okay, so the main component, as we said, of erythrocytes is hemoglobin, which is a molecule that has 2 alpha chains and 2 beta chains, so it's a tetramer, a 4-unit protein, and each globin chain, whether it's alpha or beta, has at its center a heme group. Heme is a ring of rings made of carbon, mostly, carbon and hydrogen, and at their center are 4 nitrogens that work together to coordinate with an iron atom, a single atom of iron. And they stabilize this atom of iron so that it can stay positively charged without forming rust, essentially. And so it can attract a single molecule of oxygen to it, carry it around the blood, and then release it when that oxygen is needed by a hungry tissue. All right, there are other formed elements than red blood cells, however. The second most common by number are the thrombocytes, which are more commonly known as platelets. A thrombus is a clot, so thrombocytes are clotting cells. And they're these tiny little things here that almost look like dust on your slide when you're looking at them under a microscope. If we zoom in hugely using an electron microscope instead of a light microscope, we can see that these thrombocytes really have a very small profile compared even to a red blood cell, which is a fairly small cell. And in fact, these are not actually cells. They are cell fragments. And together with the fibrin proteins that we talked about earlier, fibrinogen becoming fibrin, the platelets are a major component in forming blood clots. All right, not much more to say about platelets right now. We'll get to their function in blood clotting in a future lecture. Finally, there are the white blood cells. These are known as leukocytes, and when you see them under the microscope, they do not appear white. They appear kind of purplish. That is because we don't look at cells under a microscope without staining them first, and the stains that they pick up, their nucleus tends to pick up a bluish-purple stain, and then the rest of the leukocyte picks up different colors depending on what type of leukocyte it is. We divide our leukocytes into a couple of categories. There are granulocytes that have smatterings of granules in their cytoplasm, and there are agranulocytes that do not have noticeable granules in their cytoplasm. That does not mean that granulocytes are all related and agranulocytes are all related. It does actually happen that the 3 granulocytes are related to each other, but the agranulocytes could not be more different. Now, to compare, this is about the size of a red blood cell. Okay. Granulocytes tend to be about twice that big. And they vary largely in appearance, mostly based on their nucleus. So I'm going to sketch the nucleus as being purple here. If you have more than 2 lobes in your nucleus, okay, we call that multi-lobed. If you have more than 2 lobes in your nucleus, you are a neutrophil. At this point, you get a smattering of fairly pale granules in the cytoplasm as well. Okay. These granules are pale and small. If we look at the eosinophil, its nucleus often resembles sort of one of those neck pillows you might wear when you're traveling. This is a bi-lobed nucleus, and it has noticeably larger, though not giant, red granules. This has been compared by at least one of my students to, it looks like the cell has chicken pox. Okay. They're larger than the granules of a neutrophil, and they're a deep crimson color. A basophil has a bilobed nucleus as well, but it is harder to see because the basophil has giant blue granules that often obscure the nucleus. Okay. And it is the appearance of those granules that gives them names. There's a 3-part stain that we use in staining red blood cells, or staining blood cells rather. There's a basic component that is blue, so those are called basophils. There's a acidic component called eosin that's red, so these are called eosinophils. And then the third, more pale component is the neutral stain, and so those are called neutrophils. Monocytes are even larger than those cells, and they have a very large, often sort of C-shaped nucleus. They might have some very pale speckles in the cytoplasm that might confuse you into thinking they had granules, but they're not. They're not granules. They just are sort of speckles, artifacts of the way the cell is stained. And then the lymphocytes are typically the smallest of the leukocytes, maybe almost the size of a red blood cell, and they have a round nucleus. They have no speckles, no granules, and they have a round nucleus that takes up most of the size of the cell. Okay. Now, what do these cells do? Neutrophils are bacterial phagocytes. They eat bacteria. And if you see a large number of neutrophils in a person's body, you would naturally think that that person is responding to a bacterial infection or infestation. Lymphocytes are specific rapid immune responders. They may respond to bacteria or to viruses or even to fungi, but they must be trained before they can act. They have to be they are specific to one particular virus or bacteria, and they have to be induced to respond the first time. This is why immunization, or vaccination as it's sometimes called, is so important, is that your lymphocytes are much more effective the second or third or fourth time that they've seen a particular virus or bacteria. They're very, very ineffective the first time they see that virus or bacteria. Monocytes are chronic disease process fighters, which is why the chronic disease known as mononucleosis gets its name from monocytes being present. It's actually not caused by monocytes, it's caused by the Epstein-Barr virus. The main function of monocytes, though, is to become macrophage. A macrophage, phage, as in phagocyte, and macrophage means to eat. Macro means big, so these are big eaters. Okay? Doesn't mean they eat big things. It means they are big cells that go around eating invaders and debris and various other things. I like to think of macrophage as being like Roombas of the cell world that just wander around your tissues eating stuff that shouldn't be there. Eosinophils are triple function cells. They are parasite fighters. So if you have an elevated eosinophil count, it might be indicative of a parasite, such as a tapeworm or a giardia in your gut, or a parasite like the one that causes malaria in your blood. They phagocytize allergens and antigens, okay? And this allergen function makes eosinophils known as allergy cells. If you have a high eosinophil count and you've ruled out a parasite, or you just have reason to suspect that it's more of an allergy process, high eosinophil counts often cause allergies. Basophils are what we call good inflammation promoters, what I call that. They release histamine and heparin and prostaglandins in tissues that need to undergo an inflammation process to help clean a wound, for example. However, they can also be involved in bad inflammation, such as allergies or chronic inflammatory diseases like colitis or arthritis. Okay, now this is the best time to mention something called the hygiene hypothesis. This came out, that should be an IE, not an EI, whatever. This hypothesis came out when I was a graduate student, so it's been around for a while now. But as far as I know, it's still considered a hypothesis and not a proven theory. The idea is that there's a large amount of evidence that indicates that the reason we have allergies, the reason we have chronic inflammations that attack our own body, is because our immune system gets bored. And we didn't have these huge problems with allergies and autoimmune diseases nearly as much back when we all lived on farms and were exposed to all sorts of uh, all sorts of dirty stuff all the time. Okay, um, viruses that were carried by our chickens and our cows and our sheep and whatever else we were farming. Um, Stuff that was in the dirt that we were constantly throwing around as we did farm chores, and uh, our body is built for that, and our immune system is built to expect that kind of exposure to bad stuff. Now that so many of us live in sterile suburban environments where we're not constantly being exposed to pathogens, our immune systems are getting bored. And so they're attacking peanuts or strawberries um, wheat gluten, or they're turning inward and attacking our own pancreas or our joints for arthritis, or our own or the healthy bacteria in our colon for colitis and Crohn's disease. So the hygiene hypothesis was first published in an article titled "Let Them Eat Dirt," and it was aimed at pediatricians to try to encourage parents to let their children actually play in the dirt more. Get outside and go for a hike in the forest and let the kids actually pick up leaves and stuff them in their mouths. If you're not going to go to the forest and you don't live on a farm, at least let your kid play on the playground and throw dirt around and get muddy and put some of these germs in their mouth and expose their immune system to things that it should be exposed to. Okay, finally, this is a condensed version of the exercise that I'm posting online for you to look at some differential white blood cell count results. Okay, these are fabricated results. I made them up myself. I don't have actual patients who I stole these from. But they show you some profiles of patients who are going through certain disease processes. And if you look at these and circle the ones that are out of range, you should be able to come up with a sort of preliminary diagnosis, or at least a set of rule-out conditions for each of the people, and figure out what they're going through. Okay, next up is going to be a lecture on hemopoiesis and the life cycle of blood cells, and then another lecture on hemostasis and how we stop ourselves from bleeding and form blood clots. Thank you for your attention. If you think you learned something, leave me a like and leave any comments you like in the comments below.